Today, we will be talking about epinephrine. To give a quick summary of epinephrine, it is another term for adrenaline. It is a hormone that is synthesized in the adrenal glands and releases into the bloodstream. Epinephrine is used as a sample messenger to trigger the release of glucose from cells in the liver. Epinephrine is an important cell signaling molecule in the fight or flight response. Strong emotions such as fear and anger cause epinephrine to be released into the bloodstream, which cause an increase in heart rate, respiratory rate, muscle strength, blood pressure, and sugar metabolism. In some tissues, epinephrine acts as a neurotransmitter conveying signals between adjacent nerve cells. Now we will be talking about a ligand. It is any molecule that binds to a receptor site of another, usually larger, molecule. Epinephrine is the ligand. It is also known as adrenaline, which is released by the adrenal glands above the kidneys in response to very stressful stimuli. Acting as a ligand, it binds to its own receptor displayed on the surface of a variety of cell types through the body. Now, let's look at an adrenaline molecule.
Boom.
And another one.
Boom.
Now, we will be looking at a protein receptor. A receptor is a protein molecule that receives chemical signals from the outside of the cell. When such chemical signals bind to a receptor, they cause some form of cellular tissue response. Epinephrine binds to beta-adrenergic receptors. On the liver cell, G proteins on the inner side of the cell membrane are activated. Each G protein is composed of 3 subunits. Now we will be talking about a transduction pathway.
A series of events that converts the signal so the target cell can respond.
In epinephrine, adenylyl cyclase catalyzes the reaction of ATP to CAMP, which is a secondary messenger.
The CAMP messengers that are produced will attach to the regulatory portions of the protein kinase within the cell.
The regulatory portions attached to the CAMP of the protein kinase break away from the catalytic portions of the protein kinase.
ATP then binds to the free catalytic portions of the protein kinase, also known as phosphorylation.
The catalytic portions attached to the ATP attach to the phosphorylase enzyme within the cell.
ATP from the catalytic portions bind to the phosphorylase enzyme.
Once these steps are accomplished, the cell can respond.
Now, moving on to a cellular response. It's the target cell response to the signal. In epinephrine, phosphorylase is activated and begins to break down glycogen into monosaccharides, also known as glucose. Glucose moves outside the cell membrane to be used throughout the body. Epinephrine causes an increase in heart rate, muscle strength, blood pressure, and sugar metabolism. Now, moving on to cell signal. Epinephrine is a type of hormone, and it helps in fight or flight response.
We will be talking about the mechanism of epinephrine. Once epinephrine has reached the target liver cell, it is deceived by an adrenergic receptor protein. Because it is a ligand, it is unable to cross the membrane on its own. This is known as reception because the receptor detects to signal epinephrine.
The protein undergoes a conformational change due to epinephrine. Intracellular signal activated the G protein. This is known as transduction. GTP replaces GDP once it changes the receptor.
This activated the G protein, and then this intracellular that was now off is now on, and it goes on to the next protein in the cascade of events. During transduction, the activation G protein signal with GTB binds to adenylyl cyclase. Adenylyl cyclase then obtains ATP energy, which is made in the cell, and converts ATP to cyclic AMP, also known as CAMP.
Once CAMP is made, it acts as a second messenger that then activates protein kinase. Each protein kinase has a regulatory and catalytic part. The CAMP will bind to the regulatory side of the kinase, and the catalytic part will be phosphorylated. ATP then travels towards the catalytic portion of the protein kinase and activates it. The activated catalytic parts will move towards the phosphorylate and drop off a phosphate group.
This activates phosphorylase. The activated phosphorylase is then able to break down glycogen, which is a chain of glucose molecules. Once the phosphorylase breaks down glycogen, glucose is then released from the cell. This is the response to the signal transduction. Thank you for listening to our epinephrine presentation. So it is also known as epinephrine binds to beta adrenergic
Portions of the protein kinase. Protein kinase. Oh my god.
And then this intracellular, wait, I don't know what I'm saying.